Hi, so I'm here with Dr. Kleopa and Marina Stavru from the Cyprus Institute of Neurology and Genetics. They are two investigators who are working on a CMT1A project for gene therapy that is funded by the CMT Research Foundation. This is a really exciting collaboration for the Research Foundation for a number of reasons. One, Dr. Kleopa is really seen as a leader in gene therapy or the development of gene therapies for CMT. And bringing him into the 1A space was something that we're very excited that we can do. And then Marina, it's so awesome having you as a PhD student and having CMT and this 1A gene therapy project being the focus of your work and your degree and your PhD. So thank you to both of you for your commitment.
Thank you. It's
So
a pleasure to be able to connect with the community and with you.
thank you. Thank you. Can you tell me a little bit about the project that you're working on and what makes you excited about it?
Yeah. So first of all, CMT1A is probably most patients know that, but it's the most common type of CMT. So it's more than half of all CMT patients have this 1A form. So it's really exciting and really important to work on this and to try and find a cure for this particular CMT type. It's very exciting because we were able to transfer a lot of knowledge that we have acquired over the last 10 years in our effort to get gene therapies to the peripheral nerves with a particular twist that will address the specific problem that patients with CMT1A have. So in this CMT disease, we have what we call a gene dosage effect. So because a particular gene for the encoding of the protein called PMP22 is duplicated, so we have two copies in patients instead of one, there is excessive amount of this protein in the nerve. And based on a lot of research that many people did over the past 20, 30 years since the discovery of the cause, it looks like this excessive amount of the protein causes disruption of the regulation and of the well-being in the nerve, and in particular in the myelin, so the insulating sheath formed around the nerve fibers that is necessary in order to conduct all the messages when we want to move the muscles or when we receive the input from sensation on the skin or elsewhere. So we need to have these nerve fibers. So there is a demyelination, and this is directly related to the abnormal amount of this protein. So the whole idea in this project is to find a way to reduce the amount of this excessive protein in the nerve. And we have to do that in this particular cell type that is responsible for forming the myelin and is also known to produce most of this protein, known as Schwann cell. So this is the type of cell that is affected mostly by the disease. So in this project, we have developed a viral vector treatment to introduce a genetic therapy into these cells that will directly reduce the amount of the protein produced. And so we have demonstrated that with particular DNA molecules, we were able to target this gene and reduce the amount of the protein, first of all. And then we used an experimental model that produces excessive amounts of the human protein.
Okay.
So it essentially reproduces the phenotype of the neuropathy in patients.
Mm-hmm.
And now we're treating this model and we're evaluating various outcome measures to see whether this animal will be able to perform better in motor testing and whether the pathology in the nerve will improve and the nerve conduction that patients know from visiting the neurologist will become faster. So we use many outcome measures to demonstrate a proof of principle that this type of genetic therapy can really help to fix the problem.
So is it fair to say that the viral vector that you're using is a delivery vehicle of that gene silencing tool, and that that viral vector can be used for many different diseases? It's more like the bus that's taking the passengers where they want to go. And you're building the passenger, which is the gene silencing tool, and then you're pairing it with this viral vector that's going to get it delivered exactly into the peripheral nervous system to help people with CMT. Is that fair?
Yes, yes. So we are already using this type of vehicle for other applications, for other type of CMTs. So we work on 3 different types besides CMT1A. So it's a vehicle that has been tested. That's what I was explaining before. So
Yeah.
we know when we inject it, where it will go around the body and whether it can cause any side effects and what type of side effects. So it's a well-known vehicle. We just design the content of it that will produce the therapy in the cell. They tested many different molecules, and we selected 2 of the most promising ones. And then we had to package them into the viral vector, and then we do the testing in the disease model. First of all, we have seen that one of the molecules works really well in the model, so also in vivo, so in real life, and reduces the amount of the excessive protein. So that was confirmed. And now we're already treating diseased animals with this vector and comparing them with animals treated with the fake vector that doesn't have this therapeutic DNA in the capsules. So, and we're already comparing the outcomes. So, and we also, another important aspect in the project is that we included both early and late treatment. So we have 2 experiments. In the first one, we treat as early as possible in the life of the animals. So that would correspond to a younger patient if we treat humans. But we also treat at a later time point, at an older age, to see whether that can also be helpful. Because we know CMT in general and CMT1A in particular are progressive diseases. They start in childhood and they progress throughout adult life. And so it's important to know whether we can have a benefit from treatment both in younger patients and also in older patients. Not elderly, but older. So that's a very important piece of information. And the other thing that we included in the design of the project is that we also do a toxicity checkup. So we have another group of experimental animals where we put the vector in a healthy animal to make sure we don't cause an excessive reduction of the protein.
So I really want to talk about next steps for the project, but before we do, I know one of the questions everybody has when we talk about using a virus to deliver gene therapy is it safe? And I know you're talking right now a little bit about the toxicity, but if you could help everyone who's watching understand a little bit about the safety of injecting a virus. I mean, right now viruses are on everybody's mind.
Yeah.
And we're trying to avoid them as much as possible. Why would you deliberately inject one into your body? Can you talk a little bit about that?
Yeah, so I mean, this is a recombinant virus. It's not really pathogenic for cells, but there is, I mean, some people may have antibodies because they went through these viruses like a common cold virus. And so they may not respond fully to treatment with this virus because they have neutralizing antibodies already. but it's a small percentage, and this can be screened before giving treatment. So our approach is to inject the virus intrathecally. So it's like a spinal tap.
Okay.
It's similar to what is now being used for spinal muscular atrophy in children. So we deliver the vector directly into the nervous system near the roots of the nerves. And this means that we can reduce the amount of the vector, as opposed, for example, if we had to inject it intravenously,
Mhm.
because there you get more distribution in tissues that you don't need
Mhm.
throughout the body. So the injection method, the amount of the vector, and screening for the neutralizing antibodies are some of the aspects that really increase the safety of the whole process. And also, we expect that this will be one-off treatment. So it's not a repetitive treatment. You just give it once.
And so, the studies have shown that if we reduce the levels of the protein, at least in the mice, we get the reversal of symptoms, or, and in some cases, the stopping of the progression of the disease.
Yeah,
So,
and there is now also some good data from skin biopsies in patients that do show that there is a correlation between the amount of the protein and the severity. And so we also have data from patients. And it's also a very important field that also requires the contribution from patients. So the whole biomarker field.
mhm.
And so we use blood samples and maybe skin biopsies. And also now we have new outcome measures like MRI of the limbs to really show the cause of the cause and the clinical course of the disease in patients. Because at the end of the day, we need to treat patients, and we will need more elegant and more sensitive markers of the response to the treatment.
So, one of the things I know is, as a person living with CMT1A, is the huge importance of patients getting involved. I don't know if you can see it, but I have my scars from my skin biopsy. So I've participated in that study as well as in the MRI study.
Okay.
And continuing to go to the clinic is something that's really important to me. I feel is a responsibility of the patient, because the more patient data that exists, the more robust the clinical trials can be and the faster we can have approvals. Is that a fair statement?
Definitely, yeah. I mean, it's really crucial for patients to be known and follow up at regular intervals in the specialized centers, because they understand the disease better. They can, collect data that will be very useful in the future, and also samples. And we're not talking about any invasive stuff, really. It's just part of the routine evaluation, but if it's carefully collected in a uniform manner, then it can be very useful resource in the future. Last week I had a patient with CMT1A, and she's a violin um, player. I mean, she's 24, and she really has weakness in the hand, but she keeps playing the violin. She's graduating next year, and I mean, this patient really wants to have a treatment. Or I have seen a 30-year-old patient who goes running and wants to keep running. And when you feel your capabilities slowly going away, I mean, that's something that motivates us even more to really work harder. And we have communication with patients from all over the world, really. And so there is a lot of support from the patient, just from the moral point of view. But at the end of the day, it's also a matter of funding for research, even if it's not a big amount.
Again, as someone who has CMT, to have partners like you who are so committed to this and who are really committing so much of your life to finding solutions for me and for our community, it just means so much to us. So thank you.
Thank you for supporting us.
Yeah, you're welcome.